that we can try to interpret this.
Welcome back for the next session of our workshop. Our next speaker is Okun, who did his PhD in theoretical computer science and works at the intersection between systems and computational neuroscience, investigating the organization of neural populations, many people here, in the primitive cortex and related brain structures. And today he's going to present a talk about population-wide distributions of neural firing rates and its modulation by brain state transitions.
Great. So thanks a lot for the introduction and the invitation. Great pleasure to be here. Thanks everyone for taking the time to attend. So this talk will be a bit more experimental than the previous two. Okay, so we are talking about large-scale neuronal recordings. Right, so here's example of one, so with the neuropixels. So these are neurons, perhaps with some behavioral variables, like in projectiles. In this case, it's the pupil area of our mouse. So I think commonly when we think about modeling it, we consider summarizing what happens in time, as we've seen in some of previous talks. Here you see one very simple example where we literally just sum across all the neurons and get the population rate. But this talk will be actually about the other dimension of the same matrix, summarizing this very simple way, when we just sum average across all the time rings. And for each neuron, we simply get the mean firing rate. And as we've seen in the previous talk, this marginal actually is important for modeling as well. But I think I'm interested in this because it's quite a basic quantity. So I'll have the talk will have two parts to it. In the first one, I'll just tell you about what we know about the distribution of firing rates in the cortex. Surprisingly enough, still quite an open question. And in the second part, I'll tell you about how this distribution changes when the brain state of the animal changes. Okay, so let's start in the beginning. So surprisingly enough, until fairly recently, something like 15 years ago, we even didn't know how the firing rates are distributed across the cortical column. And the reason was, so there were lots of nice recordings even back then, and if you will remember, but most of them were done with this single microelectrode tetraroots, and by their very nature, so these devices, the experimenter advances them until she or he finds a sweet spot, which means lots of big spikes, and records in those places. But the problem is that those sweet spots are by definition biased. So there was this nice and quite well-known review that likened the situation to the dark matter in astronomy. So they suggested that actually most of the neurons are these dark, silent ones that we don't record from and don't know what they're doing. And interestingly enough, it was quite influential, and I think many people still think this is the case. So just half a year ago, I had quite an interesting Twitter chat with Rodney Wojtek. So he's based in California. His main interest is EEG. But so it all started when he posted this on his Twitter account. So he said most neurons rarely fire action potentials, less than 5 spikes per minute. So we can't really hear or see them, and he both loves and hates this function. So is it actually true? What's going on? So once it was realized that the question exists, so experimentally started addressing it. And I think still the best data that we have comes from juxtacellular recordings, meaning that instead of these microelectrodes, experimenters take glass pipettes and lightly insert them into the cortex and attach them to the membrane of the neuron, irrespective of its firing rate, and then measure what they look like. So the first paper that reported these findings by Informatics et al. So that's what they found when you plot it on linear scale. Indeed, you see that most of the neurons have quite low firing rates. But quite interestingly, they also observed that if you take the very same data and replot it when the x-axis is log scale, you get a nice Gaussian. In other words, the distribution is what is called log normal. Then a few years later, some theoreticians, so Warren, Mark Humphreys, and Christian Machens, took some extracellular data sets, which I suspect they thought are less biased than the other ones. And you can see here, observe pretty much the same result. So you see that most of the neurons on linear scale have quite low firing rates. And interestingly, so they still say that for quite a few of these, log normal is the most appropriate model, but for quite a few of them, it's either gamma distribution or an exponential, which is, so exponential is a special case of gamma. Now, this is quite important in some sense, because it actually tells us that our usual measure of firing rate, spikes per second, is actually cannot be, in some sense, a natural unit of measurement for firing. And the reason is the following. If the distribution is log normal, so that's how it looks like on linear scale, but if all we do is just rescale the x-axis, we get our nice Gaussian bell-shaped curve, but it just doesn't work with gamma, and in principle lots of other exponential distributions. So here we don't go back to zero, and if we just rescale the x-axis, we don't get anything that looks bell-shaped. And there is no way around it, but to consider how the probability density of the log rates looks like. And then indeed we get our bell-shaped distribution back. So to the extent that you believe that the firing rates need to have this intuitive bell-shaped distribution, there is no choice but to look at the log rate.
Correct. Is for the gamma PDF, are you defining log rate as some shape, like scale parameter inside the gamma? Because why is spikes per second on a log scale not log rate?
Well, because the reason is that this distribution just doesn't go back to zero.
No, but you plotted two lines. I'm just trying to understand what the difference between the second column and third column is.
So here
is
it's
the
quite
parameter,
literally,
the shape, the scale parameter of the gamma.
So quite literally the log transform. So it's not the same thing every plotting the PDF and considering the PDF of the log rate. Just mathematically, those are two different topics.
Okay.
So this mathematical observation and experimentally. So what about this distinction? Are the distributions gamma or log normal? So for a review that we are now preparing, we revisited this question. So first looking at the some juxtacellular data. So as I mentioned, we believe that this is still the most reliable we've got from this paper of Dan O'Connor. So he was one of the authors of this original black matter review paper. So unfortunately, when it is plotted this way, with spikes on the y-axis, you see that most neurons are close to zero, but hard to see. So ideally, it would be on log scale. But fortunately, you see some circles here are purple, and these are neurons that they called silent neurons, so neurons with rates below 0.01 spikes per second. So that's roughly one spike every couple of minutes. But you see that most of them are actually not silent. Fortunately, they also summarize their data using this Lorentz curve representation. So here the idea is to sort all the neurons from those that spike the least to those that spike the most and plot the contribution to the cumulative fraction of the spikes. So purple includes all the neurons, black includes all the neurons except for these silent ones. So this representation originally comes from econometrics, where you would plot the distribution of wealth, ordering all the people from the poorest to the richest. So communism, a perfect one, would be a diagonal, and obviously the closer you are to the bottom right corner, the less equal the society is. So the neuronal society is quite unequal, so some numbers here mean that the bottom half of the neurons contribute just perhaps between 2 to 5% of all the spikes on one hand, and on the other hand, the top 10% of the neurons, they contribute half of all the spikes. Okay, so then can we treat it with gamma or log normal? So the gamma fit is really nice, so you can see that the dashed line fits this Lorentz curve very nicely. Log normal actually doesn't. So if we fit it well on one side, it doesn't fit on the other, and vice versa. And even the intermediate one, which is closest overall, it doesn't fit quite as well. So the conclusion is that with this data set, which to my knowledge is still the best we've got, gamma fits better. Then we also considered some neuropixels data, so that's the best I think we have now with silicon probes, both our own and open data from Allen Institute. And here again, quite consistently we saw that gamma is better than log normal, or if indeed we use log rates, just means that log gamma is better than the normal fit. And the reason, and it's quite nicely seen here, so it's our own data from the frontal cortex, is that the actual data is skewed to the left, whereas normal distribution obviously is symmetric. And we have seen it all over the place, so with Allen data, so these are individual recordings. We've seen it in the thalamus and the visual cortex, different parts of the hippocampus. So it's quite a consistent finding that shows that many neurons indeed have no firing rates. So to summarize this first part, so basically I told you that the firing rates are logarithmically scaled, so that we know for sure, and one can even argue that log rate is perhaps the natural unit measurement rather than spikes per second. Based on the more recent and presumably more reliable data, gamma distribution seems to provide a better fit than the normal, which was suggested in the original publications. And so what about going back to this issue of dark matter and the question raised in this read of So if we take this as the best estimate we've got so far, here are the numbers. So about 15% are silent, so literally silent, don't fire at all, or just have really, really low firing rates. The rest 85% are gamma distributed with these shape and rate parameters. So this implies that about a quarter go between 0.01 and 1 spikes per second, 35% between 1 and 10, and the other quarter 10 spikes per second even more. So some words of caution about it. First of all, this is a mouse, so the human brain might be different. This particular area, so barrel cortex. Again, other areas can be different. And then one could argue that maybe juxtacellular recordings have their own biases. So the jury is still out, hopefully with some imaging methods that will get to the level of having single spike resolution in the next decade or so, we'll get the complete answer. But these numbers imply actually that this estimate of most neurons go at less than 5 spikes per minute actually is not correct. So if you calculate it, you'll see that something like 20% of the neurons do that, but actually most, so, and you can see it here as well, have quite reasonable firing rates. It's just that the distribution in space in the cortical column is, of course, non-uniform. So in the top layers, we have more of the slow neurons, and if you want to find those with large spikes, because the neurons are bigger, and also higher firing rates, you need to go deeper into layer 5. And that's one of the reasons that experimenters have to search them. Yep.
So just a clarification, I think I got lost in the details, but like, what is this stimulus-driven activity?
Yeah, so great question. That also leads us to the second part of the talk. So this is on average. So imagine that you measure for a few hours, and that's what you will get. And surprisingly enough, unless you drive one particular neuron by a stimulus that it really, really likes, the firing rate of a neuron doesn't change all that much. So a neuron that in one condition goes at 15 spikes per second, another one might be 12 or 18, but it won't go to 0.1 and vice versa. A neuron that likes spike at 0.1 will not all of a sudden go to 10 spikes per second. Yes, please.
No, I don't have questions.
Oh, okay.
Yeah, you find these neurons that spike, have low spiking rates in outer layers?
So you find more of them in layer 2-3 and layer 6. So here you see a lot of them. Here you see quite a few of them in the middle. Layer 5 usually find this. Yes, please.
And do you know the cell types? So for example, there's some empty neurons.
Yeah, so that's a great question. I think here they didn't. Yeah, so of course now that's something that experimentalists need to tell us. Yeah, but general interneurons indeed on average fire. Yeah, so this question is when we take everything together.
Yeah, it was the same question.
Oh,
So
okay,
it's
great.
entirely second.
So I guess I'm slightly
Struggling a little bit with the log rate issue, and that's partly because of the counting time. Right. So if I chose a small enough bit, such that a 1 millisecond bit, that I'm going to have spike rates that are either 1 spike per second.
Sure. Yeah, 0
So,
and 1.
yeah,
So here, the
so-
labor example
yes.
is talking about thousands or many minutes at least.
Okay, so you're counting bit. You're talking about the number of spikes produced over a very long time.
Yeah.
Okay.
So it's definitely not on this time scales of
So then this issue of stimulus conditionality becomes quite important. So you said that it won't change its firing rate very much.
yes.
What exactly does that mean? Does it mean that if I show one natural movie and I go for an hour, I get 15 spikes a second, and now the natural movie for an hour, I get 12 spikes a second? Or does it mean if I look inside that natural movie and I line it over multiple presentations, then everywhere I still just get 12 to 15?
So I think what I meant here is exactly the first interpretation, one natural mover versus the other. Of course, like I said, if you drive a very specific neuron by a stimulus that it precisely likes, you can make it fire quite a lot more. But that's not the statistics of the natural. Okay, so that's what we saw in the first part, right? So how firing rates are distributed in general, but indeed there are some differences. And here I'm talking about differences across brain states. So over intervals of minutes at least, not say 100 milliseconds to 100 milliseconds as this. So first, I want to describe a simple phenomenological model. So just purely based on theory, what's possible, and then we'll see what actually is observed in practice. So, of course, the simplest scenario is when the brain state changes, but the firing rate stays exactly the same. Then the next thing First one is a unidirectional case, when all the neurons, almost all the neurons, shift in the very same direction. So say all go up or all go down. So that's how it might look like. So we have two brain states, Y and Z. Z has higher firing rate, so the distribution shifts right. So that's one option. Another option that it shifts right and also gets stretched. And that's what you would expect if the original log rate and the modulation by how much it shifts to the right have zero correlation, right? So when you add up two random variables, variance is the sum of variances, and then the distribution should get wider. Another possibility is that the distribution shifts to the right, but actually gets squeezed. And this can only happen if there is a negative correlation between log rates and modulation. So what that would mean is that these neurons on the right that already have high firing rates, they don't change much, and these change quite a bit. So these are the only possibilities. So that's the simple unidirectional case. Generally speaking, we won't have it. We'll have a bidirectional case where some neurons will go right and some neurons will go left. And that's how it will generally look like. So again, we have two brain states, Y and Z, shown by dashed and dotted lines. So we have two subpopulations, one shown by orange. So orange is the one that's upregulated when we go from Y to Z, and downregulated when we go from Z to Y. Purple, and the other way around. So in one of these brain states, these two distributions will be closer to each other, and in the other one, they will be further apart. So here in brain state Y, they are closer. So the result would be, it's a simple mixture model that in brain state Y, the distribution will be narrower, and in the other one, it will be wider. So in this cartoon, what you see is the case where each subpopulation shifts left to right, but its width remains the same. What can also change is that this orange distribution, when it shifts, it can also again either get wider or get narrower, get stretched or squeezed. So to summarize, to completely specify or characterize a change in this distribution across brain state transition, we need to tell, first of all, the relative size of the two subpopulations, say 30% go up, 70% go down, or the other way around, by how much each one of them shifts, and what happens in terms of stretching or squeezing with each subpopulation. So the questions we have given this, so a priori mathematical structure, so these are sort of biological questions we have. So first of all, consider two specific brain states, I don't know, for example, wakefulness and anesthesia. So is the structure that we observe consistent across subjects? So a priori we expect it because it presumably reflects some physiological reality, but maybe that's not the case, right? Maybe when you are anesthetized with some anesthetics, say ketamine or something, your firing rates go down and my firing rates go up, and that's because our brains are somehow different. If it is consistent, then we can ask, well, how does the structure differ across brain areas? Is it the same in frontal cortex and in sensory areas, or in hippocampus? And we can also ask, how does the structure differ for different brain state transitions, so anesthesia versus sleep, for example. Okay, so with these questions in mind, I can finally tell you what we actually found. So we started with the psychedelic brain state, first of all because that's what got us interested in these questions originally, but secondly because when we give some compound to the animal, it's quite controlled experiment where we have a clear before state and a clear after state. But then I'll tell you in a bit also about some natural brain state transitions without any pharmacology. So the experiment is quite straightforward. We put a neuropixels probe, in this case, in medial prefrontal cortex, so areas like secondary motor cortex, prolimbic and infralimbic areas. Record baseline activity for about half an hour, then let the animal get, so it leaks sweet liquid, which either has the drug or just the control, and then wait perhaps a few more minutes and record for 20 to 30 minutes while the mouse is treated with the means for mice. So what does it do to the firing rate? So in the control case, not much. So here you see the density plot of before drinking versus after drinking, and that's the actual data. So there are a few thousand neurons here from about 50 animals. In the drug condition, so overall we still see that the points fall along the diagonal, and that reflects exactly this idea that neurons cannot change their firing rate a lot. So fast firing neuron maybe will change from 10 to 8 or 12, but will not drop all the way to 0.1 and vice versa. But we see that this cloud is quite a bit wider because the drug has some of these effects. And if we subtract these two densities, we'll see that indeed in the drug condition we have less neurons with stable rates, so that's the blue area on the diagonal, and we'll have more neurons that either increase or decrease their firing rate. So what about the distribution as a whole? So if we plot the cumulative probability, we see a clear difference, and if we fit a log gamma, we get this more intuitive picture perhaps that shows us that there is this difference, so the distribution gets wider. It is highly significant and also less clear here, but shifts a bit to the left. So is this consistent across animals? So fortunately with neuropixels, we get hundreds of neurons even within individual animal, and we can estimate all these distributions not only by putting everything together, but in individual recording. So in the controls, these are the black circles, not much changes. In the drug condition, indeed, you see that the mean consistently shifts left, and the distribution as a whole gets wider. So you see that the log rate standard deviation after, when the mouse is tripping, is consistently higher than before. So what about the structure of the subpopulations, which tells us actually what is going on? Yes?
You tried to measure skewness, was it? Skewness.
You mean, yeah, the fact that it's skewed to the left.
Yeah.
So I'll show you what happens next. So not directly, but indirectly. So I didn't tell you what happens to the width even before the skewness. Yeah. Yeah. Sorry.
Were there differences in behavior of the mice in these conditions?
So there wasn't any explicit behavior. We did monitor their pupil and paw movements and didn't see too much difference. There are some subtle differences where indeed, so I can tell you more offline. So subtle differences, yes, but nothing major. Okay, so here you see the structure of the changes for the two subpopulations. So we see that about 40% of the neurons are upregulated by the drug, and about 60 that are downregulated. And again, if we fit to this empirical data the log gamma distribution, so that's essentially the same thing as probability density. And again, we see that this picture from the pool data is quite consistent across animals. So here you see the distance between the medians of the orange and the purple distribution in the baseline condition. So indeed you see that in almost all animals close to zero, but to the left, meaning that the orange distribution is somewhere to the left of the purple one, just as we see here. And after we give the drug, it's the other way around. So the orange distribution is pushed quite a lot to the right, the purple to the left, and so most of the points end up about 0. Then, interestingly, we see that, just as in this streak, that the orange distribution, in addition to being shifted to the right, it also gets squeezed. Right? So the width goes down, and it is quite consistent across animals. So all these points are on or below the area. And the purple one is getting stretched, and it's quite clear in the field as well. And if you remember, I told you that a shift to the right and squeeze can only happen if there is a negative correlation between the modulation and the original firing rate. And that's indeed what we find here. So each point is a neuron, this black line is just the running median. So what this tells us is that neurons that have high firing rates, they don't change, relatively don't change much. And neurons with low firing rates, they change quite a bit. The reasons, so you see some neurons that fall below 0, which means negative modulation, is that this data is cross-validated. So this is the noise of the measurement. And in the purple population, we see exactly the same story, but in reverse. So here the rate modulation is negative, but again, we see that the high firing neurons don't change much, and those that fire slowly, they change quite a bit. Okay, so that was example number 1 with the drug. Then we went and considered some natural brain state transitions. So for example, transition between low arousal and high arousal. So in our data, where we measure the pupil of the animal, so we just take the top 25% of the pupil area as high arousal and the bottom 25 as low arousal. And again, we can ask what happens to the firing rates in this completely natural transition between these arousal levels. So I'll show just some of the same plots that I've shown you before. The idea is exactly as before. We take the actual data feed below gamma. So in prefrontal cortex, we see the same effect of stretching of the distribution when we move from low arousal to high arousal, and that's consistent across animals. Interestingly enough, this is quite a general phenomenon. So then we went to Allen data, and we saw the very same thing in visual cortex and different areas of the hippocampus. So here I'm just showing CA1. And again, it's consistent across animals that arousal causes this stretch, but it's not universal. So for example, when we went to thalamus, we didn't see the stretch, quite the opposite. We saw that the distribution moves right, and if at all gets a little bit squeezed, although that's not as significant. So these changes are explained, again, if we consider the structure of the two subpopulations. So for the prefrontal cortex, what we find is that that's how the data looks like. So about 60% of the neurons are upregulated by increasing arousal, and 40% go down. And the two distributions are very close in low arousal state, and then they're drawn apart. So that's how it looks like when we look at the probability density. And again, this is very consistent across animals. So a general feature of a change in this brain state. And just as in the case of the drug, these two subpopulations that don't just shift, they also undergo change in width. So for orange distribution, it's actually not statistically significant. You see that the orange points fall roughly on the diagonal. But the purple population, so neurons that are downregulated by increasing arousal, this distribution gets stretched, and this is consistent. And again, we see this clear dependence or correlation between modulation and the original log rate. that irrespective of whether the neurons are up or down regulated, those that are the slow firing ones change a lot, and those that are the fast firing ones don't change all that much. So then, of course, we can do the same analysis for other areas. So we see some interesting differences. So, for example, in the hippocampus, so the structure is overall similar, but, for example, the percentages are reversed. So in prefrontal cortex, it was 40-60, here it's 60-40. And also we see that these two subpopulations are very far apart, no matter what arousal the animal is in, and again, this is roughly consistent finding. And again, we find that the subpopulation, so the orange one, it shifted to the right, but the width didn't change, and the purple one got stretched when it shifted to the left, or alternatively, when we went from from high arousal to low arousal, so this dotted distribution went to the dashed one, the distribution got squeezed. And again, so I'm sorry, I'm a bit repeating myself, you'll find a negative correlation between the baseline low rate and the rate modulation. Here, baseline means low arousal state. So, what about the thalamus? So, why didn't we see a stretch here, unlike all the other areas, hippocampus, different cortical areas, and all we saw was a shift? So, this analysis of the subpopulation essentially tells us that in the thalamus, unlike hippocampus and cortex, it's actually the case of unidirectional modulation. So almost all the neurons are upregulated by increase in arousal, and almost no neurons are downregulated. So it behaves exactly actually consistent with the upregulated subpopulations in other areas. So when it goes to the right, it's slightly squeezed. And indeed, just as before, we found this nice negative correlation between the original log rate and rate modulation. So to summarize the second part, essentially I presented you a simple phenomenological model which characterizes changes in transition between brain states through the subpopulations, the one that is upregulated and downregulated. So the overall distribution is just a combination of the two. And also each subpopulation can get stretched or squeezed. And we applied it to actual data. We saw that in several cases, so psychedelics, arousal, brain state changes. We also looked at sleep. I just didn't show you it because it's essentially the same story. So first of all, the structure is consistent across subjects. Secondly, the brain areas do differ in relative size of the subpopulations and their relative position. So these are interesting differences that indicate that there are some different physiological processes that happen in these different brain areas. So, for example, different in the case of arousal between prefrontal cortex and the hippocampus or the thalamus. But there were also some commonalities. And what we saw across all cases is that subpopulation gets squeezed only if it moves to the right, and it gets stretched only if it moves to the left, which means that in all these cases, the slow-firing neurons, no matter whether they go up or down, they experience a relatively more pronounced firing rate modulation than the fast-firing ones. And this is potentially agrees with all these ideas that the fast-firing neurons are the hub neurons in the network, or neurons that are less prone to plastic modulation, unlike the slow-firing ones. So then, of course, one can ask how all of this relates to all the other interesting low-dimensional structures that we heard about in the previous talks and we'll hear in the next ones. So I don't have any ready answers for that one. I just want to finish by some acknowledgements. So the funders. So the first part, this is something that I worked on with Dan Lappenstein. As I mentioned, for some review that we are writing. And the second part is the work of guys in my lab and primarily Bradley, who is essentially the major part of this PhD thesis. The second part, you can read it in this preprint. Thanks, and happy to answer any further questions.
So, any questions?
I think, sorry, yeah. You go first.
Yeah, thank you for your talk. I just wondering if you so the slow firing returns are the ones like with less spikes, right?
Yeah.
So if you remove those from the data set, will you still be able to see these differences in subpopulations, or all of them will?
Well, so the question is, how do you remove them? And the issue here is, that the distribution is in the model. You don't have, it's not a bimodal distribution where you have two clear peaks, slow versus fast. It's a unimodal distribution, quite close to that, right? So it's not exactly normal, it's skewed, but it's still with Gaussian. So there is no clear line which comes and tells us these are the slow ones and these are the fast ones. Yeah, so it's a continuum. Yeah.
I guess this sort of follow. I mean, at least on this plot, the brain state Y figure. I mean, is this actually data or cartoon? This is
I
Y
think this is data, but this is fit. So
okay, but in this case, the down-regulated set actually seem like they started, or in brain state Y, they are slightly more active than the up-regulated ones.
Sure.
So you wouldn't be able to distinguish them at all by their pre-regulation state.
Yeah.
Anyway.
So yeah, the claim is not that there is some difference, but just that the effect of the brain state change is to bring the time down. Not that originally they had in some ways.
Right, so the sense that they are subpopulations, that's just being defined by their response to this particular manipulation. And so I guess I don't quite see that we're learning more from that picture than we are from just how the overall distribution, like if this distribution got wider, it must have been a case that some went up and some went down. So,
sure,
yeah.
So in a sense, I agree with that. I think it's a simple way to explain this data mathematically. So biologically, of course, it will be broken down to tens of different cell types. But what it does allow us to do is to do this analysis of how separately for upregulated and downregulated neurons, how the original rate depends on the modulation. So you can do the same where you plot absolute modulation, disregarding its sign, but one can claim that it's a bit artificial.
So I think I missed, how is rate modulation defined here? Is it in log space? Is it
Yeah.
additive effect in log space or multiplicative effect in log space?
No, so additive in log space,
So
which means
multiplicative,
multiplicative in the original. Yeah, so 1 means +1 in log space, or 10-fold increase in 5 in the
So if I plotted that not in log space, would I actually have a greater additive shift but a declining multiplicative shift?
Perhaps, but I argued in the beginning that log space is the proper one to consider. And I think Christine.
I think it's very useful in sort of characterizing the marginal statistics in your responses really precisely by sort of averaging out the function, but ultimately we do care about the function. So I was wondering, have you or others looked at to which extent the coding properties of neurons vary along this axis?
Yeah, so that's a great question. I think then we should start doing the same kind of analysis with, for example, sensory stimuli.
Yeah.
So it's possible, but specifically was not the question that we asked here, exactly because with stimuli you run into the question that Manish already raised on what time scale you are considering this data.
I was
Here
thinking something
it
a little bit different in that you still look at the large time scale, you need to do the ordering of neurons, but then look very locally at individual stimuli in this case, thing that you originally imagined as a good class, what's the people and so you could in principle separate the time scales of asking people the questions and these things.
yeah, so have them done that when they bring its information
Probably have time for one last question. So, Matthias?
Anyone else? I just want to ask you, following up on that, to speculate a little bit. So you did this work on population decoupling some while back. High firing rate neurons you showed are sort of the ones who coordinate things in networks. There's other papers along these lines. But you want to speculate what that means in the context of that work, maybe a little bit, or do you? I don't know. Yeah.
In some ways, it's a bit of a contradiction because there we saw that these high-firing neurons they are coupled less or weaker than the slow-firing ones. Unlike I think some other papers that use some other measures of coupling. So there is, so one way around it is to say that these neurons must be still coupled to the population, otherwise their spikes are completely wasted. So maybe that's the one speculation. But yeah, so that's a good question, and I don't have the answers that I'm sure about.
Right. Thank you, Michael. We can go outside to have lunch, but I must warn the people with nut allergies that these lunches were prepared in a kitchen that might have trace amounts of nuts. So, yeah.